Hi, good afternoon. It's on behalf of DG ICRISAT and on behalf of ED staff and the ICRISAT staff, it's my pleasure and privilege to introduce today's speaker, Dr. Achim Dobermann. Achim is Director and Chief Executive, Rothamsted Research, UK. And I think the best introduction from my side for him would be that he was DDG Research at International Rice Research Institute between 2008 to 2014. And I think I shared a lovely relationship with him, working with him as a researcher and seeing that how he encourages young people and people who are at the mid-level of the career to go ahead with the research. As Director and Chief Executive of Rothamsted Research, he provides leadership for a wide range of research programs that aim to develop new solutions for sustainable identification of agriculture system. Dr. Dobermann has over 30 years of field research experience from all over the world regions. And in fact, one of the things that I remember is that even when he was DDG Research, ITD, he took a piece of land where he said he will do his own research, and in fact, he used to plow that land by himself. He used to have the sowing done by himself. He managed all operations by himself and he harvested that area. And at the end of the research, he wrote a blog that how difficult it is to do the research by himself, how difficult it has been. And we enjoyed seeing him doing the application of the fertilizer, and that photograph became very viral at ITD, the DDG Research applying the fertilizer by himself. So that is what Achim Dobermann has been. He received his MSc degree in tropical agriculture in 1987 and PhD degree in soil science 1991 from University of Leipzig in Germany. And he joined ED in 1992 as a soil scientist, and later on he followed at University of Nebraska as professor from 2000 to 2007. And again he came back to ED in 2007 as program leader, and then took over the leadership of research as DDGR Research from 2008 and continued up to 2014. And then he joined the Rothamsted after taking a 3 months gap. Even when he took the gap, he already had this Rothamsted director position. At ITD, one of the significant achievement of Achim was the development of the Global Rice Science Partnership Project, and that still serves as a guide for us when we are working on the second phase of the Rice CRP. He has authored or co-authored numerous scientific papers and several books on wide range of agriculture issues, and has received many awards from various academic, government and industry organizations. He is editor of International Journal Global Food Security and a fellow of American Society of Agronomy, the Soil Science Society of America, and the Society of Biology. He is a member of Leadership Council of Sustainable Development Solution Network, SDSN, of the UNSDSN, which is at unsdsn.org. Professor Dobermann is widely engaged in initiative on implementing the post-2015 Global Sustainable Development Agenda. And the personal love that I have for Achim is that I just came from headquarters yesterday night to make sure that a promise that I made to him that when he will be at the ICRISAT, I will be here. So Achim, with this, you are welcome to deliver this seminar.
Good. Well, thank you. It's good to be back at ICRISAT. I was here a few years ago when we started setting up the IRI South Asia hub for breeding here. And I've had, of course, over the years, quite a few interactions with people from here related to the CG. I don't know what it is, but my own career seems to be 7 or 8 years in the CG, then 7 or 8 years outside the CG, then 7 or 8 years in the CG. So I'm in my second year outside the CG again. Not sure what that means, but it's actually quite healthy to make this sort of change occasionally because it helps you to reflect on a few things, and anytime you get thrown into a new environment, as I have now at Rothamsted, you start reflecting on things and you also learn a few new things. So what I wanted to do this afternoon is give you a brief overview in terms of what our thinking is, what our future science strategy might be. So, because we're still in the middle of this. Not unlike a CG center, we also are a strategically funded research institute. We're not a university. We're independent. We're not a government institute anymore. So we have to, on one side, do research that is meaningful, but we're also very much driven by the demand in our case to do research that is of high quality, because the majority of our funding comes from BBSRC as the funding agency, who places a lot of emphasis on that. But what's interesting is in recent months, there is a bit of a shift in thinking there too, because there's increasing realization that, well, we claim in the UK to have the highest quality bioscience in the world, measured in terms of journal citation impact per $1 million spent, which is true. But there is an increasing realization that had that really translated enough into real impact. And so research institutions like Rothamsted will increasingly also be asked now to make sure that the work that we do gets translated faster into some commercial value, some real world impact, something else. And that's what I really want to talk about, how we can maybe in our world accelerate innovation. So innovation in the sense that it is not just creating something or inventing something, but also thinking about then the development of the next stages and how we can do this faster. Because the one thing that has frustrated me over my entire career, including in the CG, is that we seem to be spending a lot of time on science. It takes a very long time to get something done, and it also seems to take even more time to then get something adopted, or a lot of the stuff we do that never gets adopted. It frustrates me, and I think there's reasons for this, and there's also perhaps some history lessons to learn, so I'll refer to a few of these things, but I'm more interested in the mechanisms that institutions can use, also in terms of a culture change, the way scientists need to work to make things happen faster. So Rothamsted nowadays is an institute of about 500 people, roughly speaking. So we have our headquarters is in Harpenden, outside London, just outside of London. A beautiful place, very old, 300-hectare site there that we have. We have at Broom's Barn another 70-hectare field station, which used to be the British Sugar Beet Institute, which we took over in 1960, and that closed down pretty much as a research site for us with offices and labs 2 years ago, but we still run it as a field site. So if somebody needs a research station in the UK, I can offer one. So it's Mossbolt, you can move in right there. So outside of Cambridge. Okay, we can make a deal. I'm looking for some Africa opportunities. And then 6 years ago, many people don't know this, we acquired another institution at Northwick in Devon, which is another 200-hectare site where we have about 60 people plus students to do research on grassland livestock systems. I'll show that in a minute. So we have a lot of international collaboration. It's largely through individual scientist type of projects, but I'll come back to that at the end. About a £35 million per annum budget. Nearly half of that comes as strategic funding from BBSRC. We're a luxury position still compared to you guys nowadays. But we have to negotiate this every 5 years. So right now we're in the negotiation process. We have to submit our full proposal. By end of June, we will know in December whether we'll get the next round approved. And we hope that the agreement will be for 5 years and that it will be honored, but government change can change anything, as is also the case in the CG, as you have just experienced. A promise of core funding is not a promise of anything any worse anymore. So we're in the same boat. And then essentially, this is not an unrestricted funding, it's essentially a mixture of what it's the equivalent of window 1, window 2, and window 3, a bit of everything, programs, core capability, funding, and other things. At present, we have in our current science strategy, which runs until March 2017, 4 strategic programs, 2020 Wheat, which aims to deliver science that could lead to the physiological and genetic basis for creating wheat with a yield potential under UK conditions of up to 20 tons per hectare. So we do work there on photosynthesis, nitrogen use efficiency, root traits, modeling of wheat idiotypes, plant diseases, major wheat diseases, that sort of stuff. Designing seeds is a slightly smaller program where we focus on nutritional enhancement or compositional changes in crops. So this has 2 major components. One is creating oilseed crops with an improved fatty acid profile. I'll show an example later. And the other one is on healthier wheat, so dietary soluble fiber and other traits in wheat. Cropping Carbon is a program that focuses on 2 ways of improving the carbon balance of cropping systems. One is through bioenergy crops, in our case willows and miscanthus. Miscanthus not much anymore. Willows is the only program at Rothamsted where we have a complete program from crop genetics to the breeding of commercial varieties. We don't do any breeding in wheat, for example, so we're much more upstream there. But in willows we do. It's also because we house the National Willow Germplasm Collection, which dates back to nearly 90 years ago. And so we can actually do the entire range of research there, from discovery of new traits to marker-assisted breeding in willows actually too, so perennial species stuff. And that program also has a component on root systems and soil carbon sequestration under perennial and other mixed cropping systems, including modeling of those types of things. And sustainable systems, or designing sustainable systems is its full name, is a program in which we look at a wide range of things that are usually associated with the performance of agricultural systems, both arable systems as well as grassland livestock systems. Anything related from soil health, nutrient cycling, greenhouse gas emissions, integrated pest management, weed ecology, fungicide, insecticide, herbicide resistance, bee health and toxicology, movement of insects, also genetic controls for migration of insects, and monitoring of those kind of things, and quite a bit of modeling work and mathematics involved there too. So in this program is also our site at Northwick in Devon, which I mentioned at the beginning. I'm highlighting this because it is probably the world's best equipped systems research site for grassland livestock research. We focus on beef and lamb production under grassland conditions. So we have a semi-commercial experimental platform there, which actually has 3 completely isolated different systems that we manage. So each of them is being optimized in a different way, and the cattle and the lambs herds are completely separate too, and they're hydrologically separated. We have massive amounts of equipment on there to measure basically everything that goes in and out. And this was set up about 5 years ago, and it's really just now completely operational at a stage where we can start manipulating things. And the goal there is to essentially find out, okay, what's a sustainable way of producing red meat, even though everybody thinks red meat is a bad thing from a carbon footprint and whatever else point of view. We believe there are nutritional benefits of having a decent amount of red meat in the diet, but we also need to demonstrate that there are sustainable ways of producing this, and this is our key site. It's part of a global farm platform network, which has 20 sort of sister sites or partner sites all over the world that we interact with, also one site here in India, for example. So many people don't know that Rothamsted does this kind of work. So we are much more known for some of the old things, and I'll come back to those in a minute. So we are in the moment, really at the final stages of this process here, where we started about mid-year, or almost a year ago, thinking about, okay, what's our story? Where have we come from? What's going to be our new purpose? We used to have a mission and a vision statement. Nobody could remember it. It was too complicated. It was too convoluted. Now we just call it a purpose. What's going to be our purpose? How can we translate that into a new ambition and strategy that then leads to a science strategy and with some more concrete deliverables? So this part we haven't done yet. We're close to that one. And what does that mean also in terms of the values and the culture of the institution that we want, the brand that we want? That's the hardest part, because you can talk about all of these things. If you're not changing those things as well, you're not going to achieve that new purpose. So that's what we want to do. I'd say achieving this all, including the culture change and the brand and everything else, will take another 5 years. So that'll be until the end of my normal term, I suppose. So let's see. But I'm going to go back to the history, because you have to understand as an institution where you're coming from. And most people, I think, know that our history is very much related to work we did in the 19th century on soils and nutrients and other types of things. So there were two gentlemen here who played a key role. John Bennett Laws was the founder of Rothamsted. So basically he was born in the Rothamsted Manor, the old country estate of his family, and he went to Oxford, never finished a degree, but got inspired by quite a few lectures he took there. And when he came back home, he turned the finest bedroom in the old manor into a chemical laboratory, to the horror of his mother, and started experimenting. So that's how science originated at Rothamsted. He tinkered with all kinds of things, and then in 1842 obtained a patent for the production of the first commercial fertilizer in the world, the first synthetic fertilizer, if you wish, so which was superphosphate. So he had figured out that if you would pour sulfuric acid over bone meal, you would create a phosphate fertilizer that is more soluble. And he got this patent and created then very quickly a range of commercial fertilizers. You can see an advertisement, one of the first advertisements of this kind ever produced in the world. And in 1843, he hired this gentleman, Sir Henry Gilbert, because he needed a scientist to further pursue his scientific interests, but also because he felt that for his businesses it was necessary to have a scientific basis. So he was a scientist / entrepreneur. The first thing he did with his patent was to scout for a site to build a fertilizer factory, and he built the world's first commercial fertilizer factory. But that kept him very busy, and he made a lot of money with the fertilizer business, but he needed a scientist to support his own interests, as well as then also translate the thing into the fertilizer manufacturing and recommendations. So there was an intrinsic linkage right there embedded in these two people, and so it's a bit of a science entrepreneurial approach that I think in some ways we've lost a little bit in science nowadays. But they had it because there were only two people, and these two gentlemen collaborated for the next 57 years together to basically work on scientific questions that interested them. So they spent about 20 years answering the question, where was the nitrogen in plants coming from? It was debated whether it came from the air or from the roots. So they showed it coming from the roots, and therefore you could do something about fertilizer. They spent also about, I think, 10 or 15 years answering the question, where was the fat in cows or animals coming from, demonstrating that was largely coming from conversion of carbohydrates. So these were the kind of questions they were looking at. They had the means to do it. They had laws as fertilizer factory money. They didn't have to write grant proposals. They didn't have to do with the CGR consortium or having to justify their work or whatever else. They just had fun, honestly. So they ran a private institute for 57 years the way they liked it. They never asked for any government money. but they wrote huge amounts of what we could probably call nowadays fundamental papers. And what I really liked is in their approaches, they always had a very close connectivity between field research and lab and data. So they were looking at some really big questions, and they would set up some really, what we would consider rather intriguing field experiments, collect lots of samples. We still have 300,000 soil and plant samples dating back to the 1840s. And they were basically meticulously going after things, including also inventing a lot of scientific instruments. So we have a lot of these things still around at Rothamsted. So it was this approach, looking at something, a big problem that people worried about in the field or had noticed or farmers knew, and it needed a solution. It was problem-solving oriented research. It wasn't just something that came up to their mind. Some of it did, eccentric stuff. But by and large, they were answering big questions, and sometimes it took a long time, but they were meticulous about it. We're probably most known for the set of classical long-term trials, like this Broadwalk one, the wheat trial. It's just right behind my house. I live down here. Which was set up in 1843. It's the world's oldest long-term experiment in existence. We have a few more of that kind. And it's very interesting because you can see in these trials essentially the entire history of modern technology change in agriculture. So in the first 100 years, not much changed, except that there was an effect of fertilizer or manure. So the blue line is the fertilizer treatment, and the purple one here is manure. So no difference over 100 years, just double or triple the yield. Even though there were varietal changes, and really after 1900, I think, is when sort of more systematic plant breeding started. It didn't make a difference for a long time. So, but then there was further changes in the genetics. You can see the green revolution varieties coming in here, essentially doubling the yields again. But other technological changes, liming, herbicides became available. 2,4-D, one of the most important herbicides in the world, was discovered at Rothamsted as part of the war research effort by a chemist in the 1943. So fungicides came in later. We introduced a crop rotation, which added another 2 or 3 tons. So, and in 2014, we had a new record yield in this treatment, in this experiment of nearly 13 tons with a new variety called Crusoe. We still don't understand fully why. So you can see lots of technology changes contributing to this, and it's a living long-term experiment. It is not something that is dead. So we often go back in time also to reanalyze samples for certain specific questions, because we have them. But what I often ask people is, if you would have been at this point in time here, could you have predicted all that, for example? Could you, as a researcher, even imagine, making those changes, and 20 years ahead, you would be up there, This is what we're often being asked nowadays. It's not easy. I don't believe that there would have been many people here who could have imagined all that. I probably couldn't. So, but what it also gives me hope is that, it is possible to make some huge difference if we really go after them. So it's not impossible. We've done it in the past. So we are also, I think, known for a number of other things. I'm just citing 3 more examples just to give you a bit of an idea here. Ronald Fisher was hired in 1920, I think, at Rothamsted to create a new statistics department. I'm sure that everyone is using most of the methods that he has developed, and his disciples later on, Yates, for example, in your daily work. ANOVA, Latin square design, F-test, correlation coefficient sounds familiar. There's a lot more to that. So that tradition of statistics has held on to today. We still have an applied statistics group. We have a joint venture company called VSNI, which I think some of you use software too, Jenstat, for example, but also some other stuff here. So there's a long tradition. He was hired in 1920 by John Russell because people couldn't make sense of the long-term experiment data. They needed a new statistical theory to analyze these things, and the first thing that Fisher found out was that the design of the experiment was completely wrong. They weren't replicated, and therefore he came up with a whole new theory of experimental design. And within 3 years, everybody at Rothamsted was using these new designs. So we had a long history of mathematics and modeling. Howard Penman is another example. So the famous Penman or Penman-Matisse equation for evapotranspiration. Everybody is using this sort of stuff. Came out of Rothamsted. Pyrethroids, a major discovery in the early 1970s as a whole new insecticide category with less toxic side effects on non-target specimen. This one is a sad story in a way that even though we took out a few patents, we were at that stage in our history had become a government institution. We were pretty much run like a civil service institute. Nobody paid much attention to IP in those days. We never got a dime out of this invention. It could have made us self-sufficient for until today because pyrethroids are still about 15% of the global insecticide market. Well, we missed the boat completely on that one. Or the government took the money, I don't know. It never came back to us. Shortly after that, Margaret Thatcher came into power and the reductions in agricultural funding started. And Rothamsted, which used to have at its peak in the 1980s, 1,000 people, was heavily affected by this right away. So there's a few lessons to learn from that maybe too. And we have a long history in other areas such as insect population dynamics. So we've been monitoring insect populations with light traps and suction tubes. Those were also invented at Rothamsted as a design since the 1930s. We have a radar entomology group, which is probably world leading. And we have in our radar entomology archives tapes of campaigns done with ICRISAT in the 1970s and 80s to monitor insect swarms in Africa and in India. But also with Erie in the 1980s on plant hopper swarms movement in Southeast Asia. So we still have those tapes if somebody wants to look at them and can figure out how to read them, the data. But it all goes back to technology development that took place. So there has always been a big emphasis on developing new scientific techniques. So like this vertical radar for the insect movement monitoring that goes up to 1,500 meters. We can do species recognition up to that height. Or these little antennas on bees here, which we use for studying the movement of bees in the landscape in relation to bee health characteristics or whether they've been exposed to pesticides and other types of stuff. So we have a tradition of this sort of tinkering, high quality science, also in terms of methodologies, but the question is indeed now, what's going to be our future purpose? What are some of the real world challenges that we should be in getting engaged in, and how? Because everybody's asking the same question, everybody wants to be in the same boat. We often compete against each other on needless grounds, so I think there's a huge need to also change the way we work. So we went about this from various angles. We said, all right, first of all, we need to keep in mind that there is a global need for more sustainable development. So we have now these 17 sustainable development goals, and it is good that they apply to any country, rich and poor. So it's not just something that the developing countries need to pay attention to. It's also increasingly finding its way into the policy and investment strategies of governments in developed countries. I can even see first emerging observations of this kind in the UK, which is quite surprising. Normally they keep ignoring these things. So it will guide us in terms of where we also want to work. Agriculture is, of course, highly embedded in the zero hunger or sustainable agriculture and food goal, but it's pretty much spread also in aspects of it, agriculture, food and nutrition, in at least another 5 of these SDGs. So that's good, and hopefully will also lead to more funding. We also need to be aware that despite all the science that has been done, apparently hasn't been enough to keep, in our case, British agriculture competitive. This is a little article that The Economist published last year. It was called Dig for Victory, based after a popular war campaign. And they showed that total factor productivity growth in relative terms in the UK had been far, far slower compared to other countries. And that included actually a couple of other EU countries, so we cannot only blame it all on the EU, who's stifling us with regulations and subsidies. So no, there must be other factors. So they summed it up by saying British agriculture is green, pleasant, but inefficient. So when I talk to farmers in the UK and farming businesses, they don't like to hear this. Because like in any other country, there are some who are at the higher end of efficiency and they're very good farmers, but apparently we also have too many who are not really innovators in the sense we think they could be. And maybe some of them shouldn't be farming anymore, so there are many reasons for this. But one way or another, also in science, we need to do something about this, making sure that what we do is helping to make this agriculture grow. Against the backdrop of declining importance of agriculture in a rich country like this, which then of course has led during this period to an average decline of 6% in funding for agricultural research. So that's the local backdrop. We ask people in the UK, but also many international experts, a number of questions of how they see the world changing, and I don't have time to go through this, but I got 45 pages of interesting responses. It's really interesting. But when we asked the question, what do you think a UK farm will look like in 2035, the responses were typically clustered into two groups. One group of people saying, well, everything is just going to be bigger. Bigger farms, more commercial, bigger machines, more services, more IT, everything site-specific, big data, everything. But same kind of technologies still causing problems, particularly with regard to insecticide, fungicide, weed resistance. So will become even more. So, but the conceptual model was still by many in the mindsets of people, many in the commercial industry, that efficiency and productivity seem to be linked to size. Whereas we believe that maybe there is now a time for a smarter agriculture that needs to decouple efficiency growth and performance growth from size. And where is that smarter agriculture? What's the size? How do you organize it? What is the technologies, the precision type of technologies that need to actually come down in scale and also in machinery size to be affordable and usable and practical, to also do these types of more things which other people in this set wanted to do, more diversity, more mixed farms, more whatever, healthier soils. So that's for us the interesting question. What do we focus on the big commercial side, or are we focusing on the more smarter farming that leads us to perhaps a step change, a transformation in the agricultural systems? And for a research institute of our kind, that's probably the logical consequence or conclusion. So, and there's of course a lot of thinking about new technologies. I think in our context in Europe, these are probably the five technology sectors in which we expect the biggest influence and developments in the next few years. Plant and animal genetics, I don't need to talk about this. Microbiota, or in general the microbiome, microbes in soil, inside plants, in the gut, anywhere, and how we can harness those, but also manipulate those further. Digital technologies, new food technologies, and bioeconomy, so essentially new uses for biomass and biological products that could add additional value as part of a circular economy thinking. So this is growing very much in importance, and I was at the Global Bioeconomy Summit in Berlin last December, and I was actually quite intrigued by what I saw and heard there. It had been an area which I hadn't paid too much attention to before. So when we look at an institute like Rothamsted, I think the realization that we have is that we're kind of a hybrid. We are kind of a hybrid between a knowledge institute, an institute that creates interesting things, and a knowledge development institute, so an institute that also then needs to do at least a few steps with these interesting things to make sure that some of them are taken up by somebody to do something with it. That's probably as far as we will ever go with most of our innovations. We were not going to go down very far down the end in terms of product development and rollout, if you wish. That's not our position in the research landscape. We're much better placed at the earlier stages of the innovation cycle, but it doesn't mean that we need to live in some kind of ivory tower and just do the things that we're happily interested in ourselves. And so we've been thinking about a new purpose and ambition statement, and so the working definition at the moment looks like something like this. So our purpose may be simply to say we create and develop knowledge to transform agri-food systems. So emphasis on agriculture, and then we would look at this at sort of 4 aspects of our ambition. So we want to really do good integrated agricultural science. I would say again, we've lost this a little bit in recent years. So we've become too specialized, too bioscience oriented instead of agricultural science. So with an emphasis on future farming, we see that as our core business. We see the bioeconomy as an added value proposition that will grow in importance as these new technologies and advances become further developed. We would like to increasingly change the way we operate more towards an open innovation type of model, and also working much more through partnerships and sort of catalyzing mechanisms. I'll come back to that. And we can do much better also in terms of bringing our science into public dialogue and policy setting, which we have been doing in the past, but not to the extent possible. So this is just sort of a rough way, the way we like to think about this. Now, what this then means is for an institute of the kind we are, you have to be very clear about your core capabilities and strengths. So, and if we want to work across a wide range of more complex solutions for sustainable intensification, in our case related to arable and grassland systems, it is obvious that those typically require the integration of many different scientific disciplines in a meaningful manner if we want to solve complex problems. So, but an institution of any kind cannot hope to have all that. And so, roughly speaking, this is our sort of internal mapping. So the dark blue areas are the ones where traditionally we've had strengths for a long time, and probably I missed a few, but these are the ones that we need to also maintain as our core capabilities. There are some light blue ones which either have to be rebuilt or need to be built new, particularly in the digital technology side of things. But there are many of these purple ones here where we have to have strong strategic partnerships, and it doesn't matter whether these are other research institutions in the UK, university departments or universities, whether these are international partners or whether these are companies. We need to look at either one of them and say, okay, for this particular aspect of our work, who is the best partner to work with, and how can we make this work to make sure that the things that we are good at can be linked up with these other types of expertises. And increasingly we see a particular need to link up with engineering, know-how, ICT, but also some of these things here that we don't have. So this is just a visual illustration of how we want to go about this. So we have at this stage identified 5 research challenges, or if you wish you can call them probably research programs that we will probably work on, primarily linked to 3 major themes. So productivity, protection and profitability. I don't need to explain those are fairly self-explanatory, but very much relating to the aspects that we have also as priorities for the agricultural science and development strategy in the UK, but it's globally, I think, relevant. So accelerating yield and efficiency growth simultaneously, I think this is a fundamental challenge for not just crop genetics research, but also the associated integration with agronomy and the systematic use of more precise management options for that. So these things dealing with them simultaneously is an interesting challenge that we want to tackle. In our case, primarily, of course, related to the major crops that we work on, wheat, for example, but not only. The second challenge is on future agri-food systems. So the design and optimization of the next generation of systems. So that's also arable as well as grassland livestock systems, and that would also have a very strong international component for us through work we already do, for example, in China. So what is it? How do you go about this? What are whole new systems? What are novel protein sources in those? How can we design them to also measure food chain efficiency? How do we do food chain or whole system informatics across these types of things? How do you work with industry, including the food and the retail sector, in basically managing those systems in a much more coherent manner, and also tracking and monitoring things that they want down to the consumer level. So that'd be a major program for us. Challenge 3 is on real-time surveillance and forecasting, largely about this question: how can we go about now taking advantage of the digital as well as the DNA-based sensing technologies to create an Internet of Farming Things, if you wish. So networks that allow us to be, from a monitoring point of view, one step ahead of evolution of major pests, diseases and weeds, and therefore target the appropriate inventions. So not just for biotic stresses, but increasingly also for other aspects of a more resilient, a more sustainable farming strategy at a larger scale. So it's very much related to the question of devices, sensors, and combining those with the biological sciences. Challenge 4 is then building on this and other things we do to essentially create smart crop protection solutions for the future. So things that combine genetics, chemistry, and management in a new way. We're also keen, as part of that, for example, to create a global pest genomics portal for major diseases, insects, and weeds. Challenge 5 is on designer organisms, health, nutrition, and bio-based products, and I'll come back to a couple of these examples in a minute. So these are just larger programs. The yellow bubbles down here are elements of what we hope to do there as part of these programs, but also in a new mechanism called an innovation pod. So just to illustrate a couple of these yellow bubbles. So we have a lot of our work more at the strategic and basic end. So, for example, designing healthier wheat is largely about this question of identifying new genetic sources of higher dietary fiber, identifying the causative alleles with this, and then see how we can make use of those in genetic improvement programs. So this, although it refers to wheat, actually includes now also a new project we do together with Erie on rice and a comparative biology approach. So we'd like to look at this often also across different crop species. This one here is various ways of genetic manipulations that we are aiming to do in terms of enhancing forage species, grasses for example, in terms of cell composition or lipid content, so tripling the lipid content in vegetative leaf tissue, for example, with the aim to improve the forage value for grazing livestock as part of a more sustainable grassland livestock system. So this is very much discovery type of research in which we understand or hope to understand the mechanisms and also create the genetically engineered prototypes to study and see to what extent this set could then lead to actual usage in breeding programs much further down the road. We have a few programs or activities that we take a little bit further. So this is one example. So where we are perhaps at the prototype concept stage, that's almost a little bit further already. Camelina omega-3 is something that we started nearly 20 years ago. The idea is essentially to engineer the biochemical pathways to produce a fish oil-like oil in a crop species, in this case camelina, false flax, so oilseed crop. You can see the oil composition in the camelina compared to the oil composition of bulk fish oil from wild fishery harvest. So it's almost identical, so particularly the DHA and EPA profiles, which is important. So conceptually, this could lead to a sustainably grown, crop-grown source of fish oil for the aquaculture industry and other nutritional sectors, the chicken feed and even the humans. But of course, it's a GM solution. So it's 7 genes that we have in this pathways, and now the question is for us, of course, as an institution, now that we've done a successful development, how can we sort out the IP problems that usually come along with that and gain enough freedom to operate, and where and how could we commercialize something like that in an environment where in many countries besides North America and a few others, it is difficult to take a discovery like this to a commercial stage. So we're exploring various options, but that would be an example where we would like to get engaged and also in the commercialization, but probably have to do it through creating a joint venture with another investor to also basically spin it off out of the institute. So that may happen in certain cases, but it's not very often the case for our work. The science that we do will remain also pretty strongly focused on the development and utilization of new technologies for science. We've always had this history, I showed that. Nowadays, we work on a number of things that everybody is interested in. The field phenotyping, for example, this is our fully automated robotic field phenotyping platform, which is running since June last year. It's the first LemnaTech field station that was built. You can see the camera box here. It's about 500 kilos worth of cameras in there with all of the usual sensors on there. So we've built this, but we're operating it now. We're learning how to use it. We're learning how to do the data processing pipelines along with this, and essentially hoping to go to more predictive phenotyping approaches. But we really think already in terms of, okay, how can we take this technology to the next scale from, in this case, 0.1 hectare to several hectares, and to actually make much more active use in a much broader sense for genetic discovery and breeding applications. So we have not just this, we have a whole bunch of other things around this, a whole fleet of octocopters and all the other things that everybody is playing with. So this is, I'd say, the gold standard, but we have to think about the practical applications, and that requires working with industry partners, in this case LemnaTech, but I think we also need other expertise to use. And we also want to bring expertise that we have in other disciplines, in soil science, for example, in the roots, into this kind of thinking. So how can we actually build root scanning and soil health in situ measurements onto those types of plant performance platforms to get a more integrated view of the soil plant system and also the understanding of the performance of the crop as a whole, as opposed to just looking at the above ground stuff. So that would be stuff where Rothamsted would be quite keen to invest in methodology development and testing it out with the crops that we are interested in, but essentially also with international partners anywhere. And I see a lot of potential there also to interact with CG centers. Now, I want to talk a little bit about the innovation in science and how we can accelerate this. So one thing that interests me always is graphs like this here. So Rothamsted was created, well, down here, it's not even on the scale here, in the 1840s, by these two gentlemen. But you could say by the 1860s, they had already developed a lot of interesting knowledge. But when you look at the history of agricultural output in Britain from 1867 to 1985, you will see that for about 100 years, not much changed. And that's interesting, so 0.01% growth in 100 years. And that's interesting because in this period, a lot of the fundamental technologies were known already. So things like silage making as a science became known in the 1870s, 80s. The first tractor, I think, was patented around that time here too. Fertilizers became known here, and synthetic nitrogen became sort of later here. Yeah. None of that stuff had any impact. So agriculture just did not move. So, and then it started going up here during World War II, so as a response to concern about food security, because Britain had become very dependent on importing food, and then all of a sudden, World War II cut off the import routes. The German U-boats were basically presenting a threat, so there was a big plow up the country effort. But the real fastest period of agricultural output growth was actually after World War II, until about 1965, at an average rate of 2.8% a year. And that was by and large the rapid adoption of pre-existing technologies, the innovations that by and large had been done 50 or 100 years ago. It's unbelievable if you think about this. So this happened before the Green Revolution varieties came and anything else. This later on here. So it has taken a long time, and the reasons for this are outlined in the paper. If anybody wants to read this, I can send it to you. But it has largely to do with a lot of the innovations that were out there or had been invented here weren't doable, they were too complicated, there weren't machines to do it, labor was still too cheap, the food prices were too low to make it work, all kind of reasons. It just never went anywhere. So we have to ask this question in science: how can we somehow get better with these kind of cycles? This is a typical graph that you have all seen, I'm sure, of some kind. So we think it usually takes like 15 years of research and then another 15 or 20 years to reach some kind of peak adoption rate. And we always say that in agriculture that has to be like this because it's different from manufacturing industry. Everything is more complicated. We have diverse environments, high degrees of G by E and technology by environment interaction. So it can't be standardized. People are slower to pick up on things. But is that really good enough for the future? I don't think it is. And so the question for science institutions at Rothamsted, we're at this end. At ICUSAT, you're probably a bit further at this end here, or Erie. So what can we do? So we wrote a little paper on this a couple of months ago in Nature Plants because we got into this a lot when we reviewed our position in the bioeconomy research sector, where a lot of innovation is happening. And we saw actually out of this review that, well, we should actually be thinking of agricultural science as a source of innovation, not just as something that's causing problems, but we need to really embrace a cultural change in the way we operate. And how would we go about something like this in an old institution like Rothamsted, where any kind of cultural change is very slow? And it gets even more critical because we keep making interesting discoveries, like this one recently, where we have to ask ourselves the question: does this now make sense or not? What are we going to do with this? Is it, how are we going to test something like this in the real world, or are we just letting these scientists tinker away with it forever for another 20 years until everybody concludes, well, it didn't quite work? So we've recently discovered in willows, for example, that there is a new category of metabolomic compounds which could have cancer treatment value. Willows are famous because salicylate, salicine comes from that. So they're known for that. But what in a case like this, what are we going to do about it? So and then you look at something like this. All of the various ways of manipulating plant oil composition, people are already doing in different crops. And that's now possible already in various ways. Different types of either metabolomic engineering or conventional breeding. You can see, you can create things, wax esters from camelina that could have a lubricant value, or other types of things. They could have a higher feed value, like the omega-3 for fish oil. You can do all kinds of things with sunflowers. So lots of different possibilities. And the way science approaches it, I think, will the consequence is that with the rapid advances in genomics technologies, in microbial sciences, in digital technology, whatever else, we're going to be faced with a vast array of things to choose from. Things are possible that we can't quite even imagine yet, but they will be possible in a few years down the road. The whole gene editing stuff. So how do we make use of this in an accelerated innovation concept? And how do we not get lost in all this by pursuing the wrong things? And when you look at the discussion that's going on, almost every week you can find a column like this in the major journals now. This is just from Monday in Nature, putting innovation science at the heart of discovery. Success rate could be improved if we could find out how to innovate. The argument usually goes that, all right, something is wrong with the way science is going. People need to create innovation hubs. They just need to become a little bit more willing to change the way they do things. So in our world at Rothamsted, this is how we want to do that. And we'd have to see how it works. We haven't tried it yet. So we say, coming back from our story and purpose, there is a basic science strength that we must maintain. Cannot compromise that. There are key technologies and facilities that we need, and for which we need to secure that strategic funding from our mothership BBSSE. And we have research programs. So these things here need to work in a very well matching way together and make sure that we still create a high quality science output. So the stuff that interests scientists, but also our main funding agency, in this case BBSSC, so high quality publications as a key output. All right, we need this. We can't compromise on this. It's also important for scientists and their careers. But how do we then make sure that this is not the end point? That the paper is not just leading to the conclusion, well, we need to do another research program and reaching another paper. So the new mechanisms that we want to create are of this kind. So we want to establish an innovation pod where ideas can be spun off more quickly, either things that we already sort of envision or whole new things that we haven't even thought about when we started writing our 5-year plan. So things that we can try out more quickly. Some may also spin out from these sort of higher impact sort of papers or outputs. How can we create then a physical space, an ideas lab in our case, an open innovation hub where people can go for a short time, for a longer time, for whatever time it takes, to mingle with other people that they don't normally meet, including engineers, including computer scientists, physicists from neighboring universities, astronomers. I've had a few of those over already. Interesting people. They work on image processing of galaxies with high-performance computers. This is a different skill. So you can pick up a few interesting things there. How do we make this work? How do we free up our people who are tied up in these programs and being told, all right, you got to only work on this and that's it? How do we create enough space, a little bit of space for trying things and also encouraging more risk-taking and failure as part of the culture? So that's what we hope to create in this kind of mechanisms. That will require that we have partners to work with, industry partners who participate in these things and also are willing to put a bit of risk capital into these kind of things. And we also then need to have mechanisms to make sure that some of these things, if they lead to something, can be quickly spun out in various ways. Sometimes they are more of a commercial kind. Sometimes they lead to the pre-incubation of new projects, which then become to part of the new future research agenda. But a more vibrant model, essentially, that circulates. And it requires then internally, I think, some mechanisms. You have to manage this a little bit to also make sure that things move fast. We can't make scientists get interested in this if your idea only gets attended to after 6 months. It needs to be fast. You need to basically have a mechanism for ideas generation and supporting this with sand pits, as we call them, or external stimuli and bringing in people who are completely different that we never talk to. from companies as well as universities in our case. You have to capture these things in a system. You have to sieve them. You also have to have some criteria to make sure that out of all this mess that is being proposed, there are some things that actually are quite interesting to pursue. And you have to inject then the follow-up actions, and usually it means to infuse a little bit of money to get things going, but also provide advice to scientists. Okay, what can we do with this from a commercial point of view, or from a partnership point of view? So we'll see how that works, but it is essentially a practical implementation that we hope to achieve of a leaner science concept. As a website and a whole organization dealing with leaner science, if you want to read about this. So, but in simple terms, to me, it means how can we make science faster, and how can we expose our science much faster to the client who actually needs it. So essentially, early prototype testing, and a prototype may be just an idea or a concept or a piece of knowledge. So that loops very quickly through a couple of iterations, whereas normally you would sort of sit on it for years and years and years and then hope that, okay, after we've published this paper and maybe another paper, finally we are willing to come forward with it. So we need to go this way much faster. Sometimes that actually leads to pivoting, a change in direction. So we may have seen something there from that feedback, that exposure to the outside world that guides us into a completely different direction than we had thought of. And what it usually also means is you need to be willing to start fast, but also stop fast. So this is kind of what we are hoping to achieve as part of the science culture change. We'll see how people react to this. It'll involve new ways of working with industry. So this is one example. A number of agri-tech centers are being created in the UK right now. This is the first one on agricultural informatics and metrics of sustainability, if you wish, big data along the whole agri-food chain. So Rothamsted and Reading are leading this together with NIAP and SREC, but about 200 business partners. So we will be working on any kind of interesting data science applications for digital agri-food, if you wish. So anything of interest. There is a strong element in our strategy at Rothamsted now to expand the international collaboration, and that's also why I'm here at ICRISAT today. We have recently launched a joint center in China, a virtual center at this point. It could become a bit more physical on sustainable intensification with startup funding from BBSSC and the Chinese Academy of Agricultural Sciences. There will be next week an announcement on the February 24th, an MOU being signed between various UK organizations, Cambridge University, John Innes Centre, Rothamsted, NIAP, University of Eastern Anglia and BBSSC with DBT in India to create a UK-India collaboration in plant sciences, something like that. Yeah. So, which could lead to a joint UK-India Centre on Crop Sciences. DBT, I spoke to them on Monday, wants to work really on major crops. I'm hoping that there is also willingness to include collaboration with the international centres in that. So all of that is not clear. But similar concepts, so bringing together UK expertise from various institutions to, or basic plant sciences in this case, to application in real-world research, if you wish, or problem solving related to, in this case, India. And lastly, there is a new Grand Challenges Research Fund coming up in the UK, which I think will provide a major opportunity for enhancing the collaboration with the CGIAR. You won't be very pleased about this because they've essentially taken £1.5 billion away from DFID, which probably explains some of the DFID cuts to Window 1. So they have taken that away and put it into this new Grand Challenges Research Fund for the next 4 to 5 years, which will be managed by the Department for Business Innovation and Skills, which also is the umbrella funding. The money from there goes to Research Councils UK, under which BBSRC is one of them. They will be changed into one legal entity in 2018 called Research UK. So, but the new element in this is that this money is to be used for strategic research in collaboration with international partners in developing countries, ODA-able type of research. And so it could be very useful for doing somewhat more of the slightly more upstream type of research that would provide new innovation stimuli also into research that you may be interested in, things that are more of the kind that UK institutions are good at, and how this could be linked into things for real-world problem solving. As I showed in the Rothamsted example, we don't have breeding programs. We have lots of plant scientists and biotechnologists and metabolomic engineers, but they need some real-world problems to solve, some clever things to do. So I think that's the opportunity, and that will be built actually into our strategic funding applications in June. So that's the expectation from BBSRC that the international collaboration is a component of our strategic programs and not something else. So that's it. If any of you ever have time to stop by at Rothamsted, I encourage you to do this. This is how our campus looks like at present. We opened in July last year 2 new buildings, a new conference center here, and a new open innovation hub, which is where we want to establish that innovation type of work for our scientists, but also teaming up with small and medium-sized enterprises or any other companies that want to do flexible type of innovation research. So that's all housed in this building, which is labs and offices of the finest kind. Thank you.
So, Keane, welcome. Apologies for arriving late. Really like the messages around, accelerating the innovation cycle. When I first arrived at ICRISAT, I called for demand-driven innovation. So it's really, having that rapid feedback loop with the customer, making sure that the research we carry out has relevance to society. You mentioned, behavior change in your presentation as well, and also, the role of innovation hubs. So, from your experience with CG centers, how do you see some of this thinking translating into the CGIR? That's one question. And the second is just some initial thoughts of, you're partnering with Uri a little bit already. What do you see as some of the other opportunities with Rothamsted?
I can really only speak for Uri and as a CG center that I know very well. So I don't want to draw any conclusions for ICRISAT or so, but in my view, CG centers should very much adopt the same kind of culture. This is where things are going. And I'm also saying this because I actually believe it would be great fun for scientists to do this. Come out of your sort of defensive hole. We're often sort of too cautious. We're sitting there on our ideas and we want to pursue the science and we do it and we do it and we do it. And sometimes something comes out of it, sometimes doesn't. So I think this whole concept, if it is done right and has the necessary flexibility and also speed, it could be great fun, because you get to do what you want to do as a scientist. You get to explore something, with other people that you don't normally interact with. Sometimes you do, sometimes you don't. You gain from this, So I think you should be getting out much more than not doing it. Let's put it that way. So how you do this in a CG context, particularly in this sort of shaky funding situation that the CGRR permanently represents, is an interesting question, because you need to have a pool of flexibility there in terms of people's time and a little bit of money. But I think that's doable if you want it. But I think the more important thing is the cultural change in terms of people being willing to come out of their shell and trying these things, exposing themselves in new ways. And I cannot guarantee whether that will work at Rothamsted. I know that we have a number of people who are more adventurous and would be keen to try this, but I know that we have a lot of people who would be very skeptical and actually cynical about it often too. Oh no, that doesn't work. So you need a few, I think, good examples, and that's what we're looking at right now. We're trying to identify 7 to 10 interesting examples where we say, let's try those, to get a few, and then hopefully have the right individuals along with that, line up a few industry partners, to make it work and also learn how to do this. The key is not to make it bureaucratic. Everybody in science is stifling from bureaucracy already. So if you can create something that is less bureaucracy, more fun, gets you to something faster, explore an idea, even if you find out after 6 months it's rubbish, fine, come up with the next one. So in terms of our interactions with ICRISAT, I'd be interested in a number of areas for which I've submitted a number of ideas to BBSRC already as potential ideas for this Global Challenges Research Fund. So one is on predictive phenotyping, basically, and the whole phenotyping approach and technology. How does one really take this to the next level, and basically doing this across various cereal species, wheat, rice, and the ones that you're interested in, for example. So that'd be one. We had earlier last year developed a proposal which did not get funded for a public-private consortium with industry, and this was actually led by Lockheed Martin because they wanted to bring in technology from the defense and aerospace sector. So that's the sort of stuff I'd be interested in, where I think everybody is in need of this, but everybody is lacking a little bit the last touch of technology needed. So that'd be one area. I would be interested in areas related to our philosophy on smart crop protection and real-time surveillance and forecasting. So it has a strong sort of digital component too, but not only. So we're interested in that. For example, we hope to set up a pilot for fungal diseases in wheat in the UK and for monitoring the evolution of black rust resistance to herbicides and a few insects. So how would you go about this in a developing country setting? And what can you do with that kind of information to guide interventions faster, for example? Or the whole concept in the crop protection area of is it possible to reduce our addiction for chemicals, which is of course in Europe much stronger than anywhere else, but you have that in many other parts of the world too, towards let's say a one spray crop, as opposed to 5, 6, whatever. How do you go about this, and how do you bring the genetics, the chemistry, and the management together to really have more sophisticated ways of more durable control of major pests in our context, but also in the context of India or in Africa. So we've been working, for example, in East Africa for 20 years with ECP on the push-pull system. So that's an interesting partnership because they've done sort of more the applied side of things and now the early stages of extension, if you wish, within their limits. Whereas Rothamsted in that case did a lot of the chemistry work involved, semiochemicals and all that stuff. So where we can find those kind of bridges. And I'd also cite another one, which is soil health. So it's something that everybody is bothered about. What do we do about soils? And there's confusion a lot about this too. I think we need an entirely new paradigm of how to measure soils, if you wish, in a non-destructive way, because our classical way of collecting a soil sample, carrying it to a lab, extracting it with wet chemistry, all this stuff, and I'm a soil scientist, I've done this for all my life. The whole science behind this hasn't changed for 50 years, 60, 70 years, I would say. So we need a completely new way of thinking there. But that would be the stuff that we would be keen on from a European point of view, but also then applying it to African and Asian conditions. So these are just 3 conceptual examples of this kind. where I could see interesting linkages.
Well, you say that you are targeting, let's say 20 tons per hectare yield in wheat, and in your initial graphs, haven't we? The reason why those graphs are plateauing is it because we have reached, the thermodynamic potential? And then the second thing is you further down in your presentation, I sort of like the concept of looking at agriculture in a different way when you look at efficiency, not necessarily looking at size, but looking at agriculture that produce other services and producing food. But so isn't this in contradiction with the 20 ton per hectare? In other words, you can't make omelette without breaking eggs, and so are we not shooting at an optimal in terms of efficiency rather than total?
Yeah, no, you're good. So I'll put it in the UK or European context, and you can translate it into the developing countries the way you want. Yeah. So the UK wheat yields and for most of Western Europe have stagnated for 20 years. Not much increase, even though breeders are claiming genetic gain in their variety trials. Actual farm yields have not increased substantially. They average about 8.5 tons in the UK. All right, so that's their average. So yield potential simulations that we do would suggest that in the climate that we have, it is possible to, in some parts of the UK, to grow wheat with a yield potential of up to 20 tons. So that's yield potential. Let's be straight. We have for very, very progressive farmers who are also competing in a yield enhancement network in the UK in the last few years, who every year set up new records. I'm almost a bit suspicious about it. Last year, this winner set up a new world record of 16.4 tons or something like that. They're getting there. We get in our trials 13, 14 tons already too. So the real issue is not yield potential, although that's what our program is to a large extent focusing on. But not only, it's also about closing the yield gap. So, and that's puzzling me, because when I came to the UK, I started looking around and asking people, what's our yield gap and why is it there? And nobody seems to have a very clear answer now why our yields are only 8.5 on average and how variable the yield gap is. Nobody has mapped the yield gap and systematically understood it across the country. It's even worse in canola. Oilseed rape yields have not increased substantially in the last 30 years. So there is a complete lack of understanding of what the causes of these things are. I wouldn't say complete lack, but a substantial quantitative lack of understanding. So, and that's interesting, because you could be asking yourself the question, why then are people still investing into breeding? It seems to be just maintenance. So at the same time, we have racked up huge amount of problems in agriculture. Resistance to diseases has fallen down, or fungicides become less effective. Insecticides get more regulated. We have trouble with weed control. Largely, these problems are because we have gone away from proper crop rotations. We've become wheat, oilseed rape people, trying to fix everything with a quick spray. So we need to actually, from our science point of view, go back to the basics a little bit first and answer this question: what are the reasons for these current gaps? To then address the question, what do we need to do about in terms of real yield enhancement versus efficiency enhancement, and where is the real potential? So, but from a trade point of view, we will actually stop the work on photosynthesis in wheat, because we don't see much need for it, except I'm very keen at some point to start working on a much more fundamental question, which is C4 photosynthesis in wheat. Once the C4 rice project is ready with a bit more work, I'd like to start working on wheat. That'd be the next thing now to get going. But we will concentrate our trade research on wheat at Rothamsted on architecture for a healthier canopy and nitrogen use efficiency, which is a very difficult one. So that's the sort of the logic we're trying to get through. Thanks, Simon.